This is a production of Cornell University.
Thank you, Carl. Can everybody hear me okay? Okay. Yeah, I'll be talking a little bit about my research with malting barley today. Okay, so in 2012, there were 92 craft breweries in New York, or breweries in general in New York. Now, in 2018, we have over 400 breweries in New York. In that time frame, breweries across the United States have boomed, but the growth of breweries is particularly striking in New York State. Much of this can be attributed to the Farm Brewery Bill. This is a state bill that was passed in 2012 that provides economic incentives for breweries to use locally produced ingredients. Specifically, at the current quota, farm breweries must use at least 20% locally grown hops and 20% locally grown malts to qualify for these incentives. That quota increases to 60% in 2019, so just in a couple of weeks here, and increases finally to 9% in 2024. Currently, we're producing enough malting barley in New York to meet these quotas, even at the 60% increase in 2019. However, Cornell Cooperative Extension has set an ambitious goal of 30,000 acres by 2023 to account for the demand at the 90% quota. So if we're to meet that goal, we must increase malting barley acreage in New York. However, high quality malting barley is not an easy thing to produce in New York. This is largely due to the wet, humid climate in New York that we frequently have during our summers. There's a long list of problems that can happen. I'm going to talk about two today specifically. The first is pre-harvest sprouting, which is a premature loss of dormancy in the field before the grain is harvested, which can result in early germination when a moisture event occurs, such as a heavy dew or prolonged rain. The second that I'll talk about is foliar disease, specifically leaf rust and spot blotch. These are both fungal diseases that can cause yield loss, and spot blotch can also negatively impact grain quality. So to meet these challenges, the Cornell Small Grains Breeding Program initiated a project that we're calling Born, Bred, and Brewed in New York. And our goals are to support farm brewing by quickly meeting the needs of local maltsters and brewers who are operating under the Farm Brewery Bill. We're hoping to breed high dormancy to avoid pre-harvest sprouting, disease-resistant varieties that have high malting quality across the variety of New York environments. Malting quality also needs to be stable because maltsters and brewers need a consistent supply of malt to make consistent beer year after year. So to do this, we're establishing a 2-row spring barley breeding program at Cornell. Maltsters and brewers want a New York barley variety yesterday, so we have to act quickly. Naturally, seeing that I'm in the Sorrells lab and then I'm at Cornell, our approach is going to be genomic selection, but we wanted to make this, we wanted to hit a home run on the first try, because with this strict time limit set by the Farm Brewery Bill, we don't have a lot of time to waste in breeding. So our approach is to use a nested association mapping style design for our genomic prediction training population. For those not familiar with the nested association mapping panel, or a NAM, it's basically a collection of hapsid families connected by a common parent. And the idea here is to use the beneficial statistical properties of the NAM to increase our prediction accuracy with higher relatedness, large population size, more efficient genotyping due to population structure, ideally increase genetic variance for multiple families instead of just 1 or 2, and then also high power for genome-wide association studies. Since we're doing genomic prediction, we're not interested in the genetic architecture per se of these traits, but we're interested in adding any high effects marker loci as fixed effects in our prediction models. Even if none of these work, it's still a useful breeding population to start a breeding program. So our approach was to use the Canadian variety AAC Synergy as our common parent. This has performed fairly well for us in New York State. It has high yield, high malt quality, and high disease resistance, but does not have good pre-harvest sprouting resistance. We used 7 other lines to cross the Synergy, and we ended up with a population of about 1,300 lines, so a little under 200 lines per family. In 2017, so last field season, Two heterolocations of F3 derived F4 lines were grown and phenotyped for the traits listed there to train the prediction model. And this year, a subset of 100 lines from that training population were grown in regional yield trials. And I'll come back to that. So this principal component analysis here gives a nice indication of the population structure here. So the colors got a little washed out, but the green and orange dots there on the left are all crosses made from North American germplasm. North American barley germplasm is not very diverse. It's been heavily selected for malting quality over the past 50 years, so it's all pretty closely related. Right there at the top, there's this little yellow cluster. It's actually a back cross. We didn't realize that Newdale was one of the parents of Synergy, but it is, so we have that. And then the blue dots here in the middle, that's a European by Canadian cross. And the European malting barley germplasm is kept pretty separate from North American germplasm. So a lot more diversity there. Okay, so our genotyping approach for this population was to genotype the progeny at a higher plex than the parents. So with this many lines, we needed 4 libraries. So the parents are present in each of those 4 libraries. Each of the progeny are only present once. So the parents got sequenced at a 4 times greater depth than the progeny. We were able to align these sequences to the recently updated barley reference genome, which is pretty good. So we're confident in our physical positions of these markers. Because all the parents are inbred lines and are released varieties, I was pretty stringent with the parental filters, so removed any heterozygous sites, any missing sites, and only kept sites that I was very confident for those genotype calls. And then within family, I imputed monomorphic markers to those parents, and any other missing data were imputed with expectation maximization in R BLUP and R. The usual heterozygosity and mononuclear frequency filters within family, and then a final filter to remove any markers in very high linked disequilibrium with each other. So that gives me about 6,000 genome marks to work with. Okay, so I'm working here with single trait GBLUP models that I fit in ASRMOR. These are tested with a 5-fold cross-validation with 5 times. So there's a lot going on here. But these gray boxes correspond to prediction accuracies with the entire set, and each of the colored boxes refers to the individual families here. So there's a couple trends that jump out here. The first is that when you look at the gray boxes, these prediction accuracies for the whole set, they tend to be as high or higher than prediction accuracies within each family. This is what we were hoping to see. Prediction accuracies within each family vary widely depending on each trait. That's largely because the genetic variance in each of these families varies a lot. Specifically notice this yellow box here in the middle. This is our backcross family and has consistently low prediction accuracy for each trait and is consistently lower than the majority of the other families. The standard deviations for the prediction accuracies are quite a bit higher for pre-harvest sprouting and spot blotch. This is likely due to incomplete phenotyping for those traits. Pre-harvest sprouting is a very labor-intensive and time-sensitive phenotype, so we weren't able to phenotype that large of a population. And then spot blotch, the data was incomplete due to premature senescence because of rapid disease progression. GWAS models were fit in our BLUP with a single principal component added to account for population structure. The hits that we see for leaf rust and for spot blotch lie within regions that have been previously mapped for these 2 traits, so we're fairly confident these are real. This very large effects locus here for pre-harvest sprouting is in the region that a recently cloned dormancy gene lies in, and that dormancy gene is implicated in pre-harvest sprouting, so also confident about that. And this height locus is close to a well-described, commonly used semi-dwarfing gene used in European malting barley germplasm, and that allele is only present in the European by Canadian cross. So the marker data seems that's useful. So again, we're not interested in understanding the genetic architecture per se of these traits. We want to see if it increases prediction accuracy. Unfortunately, it seems that only adding that very large effect marker loci for pre-harvest sprouting increases prediction accuracy. However, when we take a little bit closer look at trait heritability and percentage of genetic variance explained by these loci, the picture is a little more clear. So Rex Bernardo was looking at the same problem in a simulation study in 2010, and his results somewhat mirror mine. So for traits that have low heritability and have, let's say, a GWAS set that doesn't explain that much of the genetic variance, so something like height or spot blotch, low percentage of genetic variance explained, adding the fixed effect doesn't really change prediction accuracy, which is what we see for height and spot blotch. However, for a trait like pre-harvest sprouting that has very high heritability and has a locus that explains a lot of that genetic variance, not surprisingly, that will increase your prediction accuracy, which is what we see. Leaf rust is a bit puzzling, though, because we have high heritability, and the percentage of genetic variance explained is not low, but it's not really increasing prediction accuracy. So that's a little mysterious still. When we use the 2017 training population data, though, to predict unobserved trials, which in this case are 2018 yield trials, adding fixed effects has a more significant impact. So N here is the number of 2018 yield trials that traits are measured in. So when we add fixed effects for these traits, you can see prediction accuracy increases quite a bit, especially for pre-harvest sprouting for all these traits. For something like pre-harvest sprouting, this is particularly promising. Pre-harvest sprouting, like I said, is a very time-intensive and labor-intensive process to phenotype, and we can only do this locally, but it's one of our most important traits. So having good predictions for unobserved yield trials for this trait would be of great value. Another thing we'd like to do, and we're starting a genomic selection project from scratch, is to use as few markers as possible to reduce costs. So at a number of different marker densities, going from full marker density all the way to 1% of the full marker set, I sampled 25 different marker sets of each of these densities and used fivefold cross-validation to test prediction accuracy of each of these sets. Promisingly, it looks like we can go down to about 10% of the total marker set and maintain prediction accuracy for most traits. So that's 600 markers compared to 6,000 markers. This is partially due, I think, because barley naturally has high linked equilibrium, but also its population structure helps. You see prediction accuracy tails off much more rapidly for pre-harvest sprouting, likely due to that single very large effect locus of pre-harvest sprouting. When you're only sampling 300 markers, the probability of sampling a marker in LD with that main causal locus decreases accordingly, so prediction accuracy drops. Lastly, we would like to have smaller training populations, because genotyping 1,340 lines is a lot of work. So I did use a similar cross-validation strategy in testing smaller training population sets. For most traits, we can get down to 50 to 75% of the full training population set size and still maintain prediction accuracy. However, again, we see this major drop off in pre-harvest sprouting. I think that that's largely due to the incomplete phenotyping. So I'm only starting with about 400 observations for pre-harvest sprouting. When you drop that by 75 or 50%, you have many fewer individuals that you're using there. So with these results and our regional yield trials in 2018, plus yield trials next year, we're confident that a New York malting barley is within reach. We've identified high yielding, disease resistant, and pre-harvest sprouting resistant lines, but we still don't know how they malt across environments and across years. So this is data we're waiting on, and that picture will become more clear next year. We're definitely speeding this process along, and we're hoping to identify a variety candidate in fall 2019 and get that in foundation seed in 2020. Hopeful farmer release within the next year or two. The other part of my dissertation research is using selections from this training population to initiate a genomic selection experiment. Currently completed 2 rounds of that, but validation will be this summer in the field, so stay tuned for that. I would like to thank my committee members, past and present members of the Sorrells Lab, and folks from the Bergstrom and Vian's Lab, as well as funding partners. And with that, I'd be happy to take any questions.
Any questions for Dan?
Okay.
How did you pick the parents?
Mark had grown yield trials for 2 or 3 years before I got here. And at the time, those were our best performers in the data we had. Going back, I would not choose some of those parents, particularly Mead Ale, the one that was the back cross. But
Can you repeat the questions?
oh yeah, sorry. So Margaret asked, why did we choose the parents that we did? We went on what existing data we had, which was 2 or 3 years of yield trials. But if I were to do this again, I would choose some different parents.
Yeah, I was just curious, like what you could do, because I feel like all the prediction accuracies, like what you really wanted was the pre-harvest sprouting.
Yeah.
But like that was the hardest thing to predict, at least like with lower markers and things like that. So
Yeah.
what could you do to, this is a hypothetical question, if you were to do the experiment again, is there something you could do differently to like make sure your predictions of that are better?
Yeah, so I think the biggest thing would be to phenotype it more completely. And I think repeating the question there. So the question was, pre-harvest sprouting is our most important trait, but had the most severe drop in prediction accuracy when we did subsets. I think the key there is phenotyping more. Like I said, we only phenotyped about 400 to 450 of those 1,300 lines because you have to sample them at physiological maturity, which is a very short window. So either we hire 3 times as many undergrads, which could work, or we have a small population size, or work 16-hour days, either one of those. But I was pretty happy with the prediction accuracies we did get for pre-harvest sprouting. We knew very little about the genetic control of this trait in our population and how it worked in New York. We have a much better idea of that in wheat, but these results I think are useful in barley. Yeah, it's here.
The one that was like almost an outgroup, the Tinka.
Yes.
Where was that from again? Was it phenotypically different from the other ones that were all very similar genetically, or did it look about the same?
Yeah, so the question was, there's this kind of outgroup line, Tinka, which is a European variety from KWS, so bred for continental European climates like France and Germany, which in our experience, those lines tend to perform a little bit better in New York. There's a lot of barley breeding in the UK, but those lines are adapted to a maritime climate, which we don't have here. So it's much cooler in the UK, and it can get pretty hot in the summer. So lines bred for Germany and France do better. Phenotypically, it looks fairly similar. It is a little bit shorter. It's susceptible to several diseases here, but it has good malting quality. These are all 2-row lines, so 2-row malting barley all kind of sort of looks the same unless you spend hours in the field. So yeah, it looks similar, but has a lot more genetic diversity. All right, thank you.
This has been a production of Cornell University, on the web at cornell.edu.